So according to the second law of thermodynamics, energy goes towards entropy. Things go towards disorder. But how would this work in our cells? How would we still be able to function if it's all just chaos and a big mess in there? Well, actually, our cells have all these mechanisms inside that they're able to regulate everything that goes on and all these different processes and reactions to be able to maintain order. So when we're talking about enzyme regulation, we have a bunch of different ways to do that. So if you wanted to change, say you were making a lot of a certain product and you wanted to decrease that product being made, well, you would inhibit the enzyme that takes that substrate and turns it into that product, right? So that enzyme, to inhibit it, you could use competitive inhibition. So this competitor just has to have a similar shape to the active site, and then it would be competing for that same active site. But say if there's equal amounts of the competitive inhibitor and the substrate, there's like an equal 50-50 probability that either one will be getting there. But if you want to inhibit it, you would increase the concentration of inhibitor, or vice versa. If you wanted to make more of the product, we would increase the substrate concentration. Now we can also have allosteric regulation, which pretty much means allosteric site is a site that's other than the active site. So it's not actually where the substrate would be trying to bind anyways. It's this other one somewhere else on the enzyme. And this allosteric inhibitor, or activator in some cases, comes along and changes the shape. In this case, the shape change means that the substrate is no longer able to bind, and therefore the reaction will be stopped. Or at least it won't happen fast enough to sustain, to actually work in the body. But we could also have allosteric activation, so it could be that the shape is wrong, and then an allosteric activator comes along and then changes the shape so it now fits the substrate. Then there's also, you could change the bonds on the enzyme itself. So they have these OH groups on the edges of the enzyme. So you can either add a phosphate to them, and what adds phosphates is kinases, and that's what would turn it on for some enzymes. But it's kind of confusing, but for some enzymes, adding a phosphate can also mean you turn it off. And then doing the opposite, so if it was on with the phosphate and you want to turn it off, you would take off that phosphate, and what takes off the phosphate, it is this thing called phosphofatase. And then you could also do, now removing a phosphate doesn't always turn it off, it depends on the enzyme again, so removing a phosphate could actually turn an enzyme on. It depends on the enzyme and what it's interacting with in that particular reaction. So these are all the different things you can use to regulate the reactions happening in the different pathways. Remember that reactions in our cells are not just isolated little things. It's all part of this big web of reactions, and they all interact so that if there's too much product here and you want to stop the substrate causing it, then you can go back and use one of these mechanisms to do so.